When I talk about drugging the cancer genome, I'm referring to the fact, obviously in terms of genome, that we know about 500 plus genes that are responsible for contributing to or causing cancer or prolonging it, making it more aggressive. So there you've got about 500 genes that I refer to as the cancer genome. And when I refer to drugging the cancer genome, the notion is that ideally you would want to develop a drug that worked on every single molecular target, so that whatever genetic makeup a particular tumor or particular patient's tumor had, you could come up with a drug combination that was ideally suited to that particular setup. Right now, we only have about 5% of all the genes in the cancer genome, 5% of those 500, that actually have a drug working against them. That leaves 95% dark matter, if you like, of the druggable genome. And so the message here is there's a lot more drug discovery work to do to build the full armamentarium that we need to treat cancer. I really started with the central theme that we now know that drug resistance is a fundamental problem. It's a fundamental problem in infectious diseases, it's a fundamental problem in cytotoxic chemotherapy and cancer. Now we know, we shouldn't have been surprised, but we perhaps have been surprised, that it's a fundamental problem in targeted therapy. So first of all, I discussed how this has come about. We know that there are feedback loops biochemically. We know that there's clonal evolution whereby cancer cells are unstable genetically, and that allows a fertile breeding ground to be present so that when you come in with a drug, you develop resistance through growing out clones that have resistance to that. So that was the background I talked about, and then I suggested several strategies that one could use now to combat this and to overcome drug resistance. The first strategy is a simple one in concept but hard to do, which is to use targeted combinations of drugs. So if you think about the feedback loops, when you inhibit a cancer pathway, the cell rewires, it alters gene expression, it alters biochemical responses in pathways, and therefore you needed a combination therapy that would overcome that new rewired setup. Then if you take the clonal evolution problem, you need to think about choosing not just yet another kinase inhibitor, although we do need more kinase inhibitors, but when we think about targeting a new drug target, you need to think about how will the cell become resistant to that, and how will we, if we have such an inhibitor and a drug, how would we use it clinically so as to reduce the chance of that resistance developing. What I went on to say was that we have been working in my lab and in other labs around the world for quite a long time, pioneering the notion that a target that we could hit in cancer called HSP90, developing drugs against HSP90, would actually work both on the feedback loop mechanism and on the clonal evolution mechanism. So first of all, if I just explain how the biochemical feedback loop problem might be overcome. When you, as I mentioned, when you inhibit a drug pathway, new genes get switched on, or the pathway gets activated in a different way, and you get resistance to the initial targeted drug. The advantage of an HSP90 inhibitor is, although it's not a blunderbuss approach like the old cytotoxic drugs, nevertheless it hits several different pathways in cancer and many of the key proteins in cancer which happen to depend on HSP90. So right there, by inhibiting HSP90, you block multiple pathways in the feedback loops, and that's how we think HSP90 inhibitors have already shown some activity in the clinic. Next is the clonal evolution problem, and with the reason why HSP90 inhibitors would have a chance of overcoming this fundamental problem of clonal evolution is it turns out that HSP90 is critical for the way that proteins evolve. That's true if you go back through eons of natural selection for organisms. There's many examples where when resistance develops, protein has to evolve into a new form. That new form is unstable in the cell. And unless HSP90 is present, it's hard for that protein to become to show its activity and to remain stable in the cell. So that's the first way by which an HSP90 inhibitor would just block that evolutionary process at the protein level and block new proteins evolving and contributing to that clonal evolution mechanism. In terms of practical utility with HSP90 inhibitors, one of the things we really need to do, because we are still in the early days of these drugs, we need to understand how they work, and I showed in my lecture new data that we've only recently published that shows precisely what happens when a drug binds to the HSP90 complex and how that re-engineers the complex and how the drugs actually work. And we actually show that not only is it important when you block HSP90 that you destabilize these nasty oncogenic proteins, you actually block their signaling activity. That's the biochemical switches that they have. And that was a surprising finding, so we've just shown that. But then in addition, I've shown that there are complications, because even with HSP90 there are potential resistance mechanisms to an HSP90 inhibitor, despite it being a network drug with these various desirable effects. And so it switches on the so-called heat shock response, and that's a protective mechanism. So I talked about how we ran a new data, which we've only just beginning to publish now, where you run a large genetic screen and find other genes that, when blocked, give a much bigger cell killing effect with the HSP90 inhibitor than you would just with HSP90 on its own. So that's a new approach for the future, but a longer term one, because we still have to develop drugs that work on that second target. What we can do right here and now is we can say that where we have HSP90 inhibitors that already work in the clinic, and they work in HER2 positive breast cancer that have become resistant to Herceptin, and to non-small cell lung cancer that have become resistant to ALK inhibitors. In both of those cases, if you use an HSP90 inhibitor after resistance, you can see 20-30% response rates. What I pushed very hard in my talk today is the need to move HSP90 inhibitors much earlier in the treatment cycle. It makes complete sense, because we want to block resistance and block evolution happening, not try and reverse resistance or reverse the evolution that's already taken place. And I referred to two recent publications, one with steroid drugs in breast cancer and one with vemurafenib, which is a drug that inhibits BRAF oncogene in melanoma. And in both of those cases, in these two nice pieces of work, if you use the HSP90 inhibitor right at the beginning of treatment, together with the targeted drug, you have a much bigger anti-tumor effect. It lasts longer, and the resistance is much slower to develop. So I think both of those preclinical studies really support what I'm pushing very hard now, which is for clinical trials that will work in the same way. I also talked in my lecture very briefly about another important area, which is again where we've discovered drugs. We've discovered PI3 kinase inhibitors as well as HSP90 inhibitors. And the drug that we developed is a representative of this class, and we don't completely know how to use them, because it turns out they don't have a very simple biomarker to tell you which patients to treat. It's more complex than that. And also it looks like you have to use them in combinations. And again, they show promise when combined with an anti-hormonal treatment in particular subgroups of breast cancer. And what I revealed was yet another example of resistance. This is due to a mechanism where you increase the expression of the mutant drug target, and that makes the cells much more resistant to the HSP90 inhibitor. So we need to find ways of overcoming that, probably again with another targeted drug combination. I think the overall take-home message from what I said is that drug resistance due to clonal evolution and feedback loops is the fundamental problem that we face in all of our clinical trials. We need to think hard about the underlying biology of that and choose targeted treatments that respond to the genetics and the biochemistry of the individual tumor. And ideally, those combinations would contain a targeted therapy that address the fundamental pathways and mechanisms involved in the evolution of resistance, and HSP90 would be the first example of that new approach.